Hello and welcome to MCAT Outreach by MCG. My name's Remy Alway and I'm going to be teaching you about amino acids today. So the first thing that I want you to know is that amino acids are the building blocks of proteins, and proteins do all types of various functions throughout our body. So just think about amino acids like as very important molecules in our body. So what I want you to know first is amino acids have four groups attached to a central carbon or an alpha carbon as seen here in this image. And going off of that, there's an amino group, there's a hydrogen group, there's a carboxylic acid, and then there's a R group. The R group determines the chemistry and function of that amino acid. So basically, whatever the R group is can determine whether the amino acid is basic, acidic, hydrophobic, or polar. There's 20 different types of R groups, and thus there's 20 different types of amino acids. So in the body, you kind of have to sustain like a normal neutral pH. Basically, the MCAT thinks of a neutral pH as about 7.4, anything around that. And the important thing to know is that amino acids are in a Zwitter ionic form at this pH. And what that kind of means is that there's a negative charge, but there's also a positive charge on this amino acid. So as you can see here, the carboxylic group is now missing its hydrogen, which means it's deprotonated. However, the amino group still has its hydrogen, which means it's protonated. Basically, and I'll touch more into it a little bit later, but basically this is determined by the pKa of both of these groups. So for carboxylic acid, you can think of the pKa as being roughly about 2. While for amino groups, you can think of the pKa as being around 9. So basically, if the pH is greater than the pKa, you are deprotonated. But if the pH is less than the pKa, you are protonated. So this explains why the carboxylic group is missing its hydrogen, but the amino isn't. And this kind of also goes into the R groups, and we'll touch on that a little bit later. But this is just kind of a good starting way for you to recognize. So some of the properties of amino acids, and I'll tell you which ones I think are the most important. It's important to know that carboxylic groups have a pKa of about 2, and that amino groups have a pKa of about 9. The third pKa, or the side chain, which is going to just be our R group, that's going to be determined by the R group that it is. So if it's basic, it'll have a higher pKa. If it's acidic, it'll have a lower pKa. Given the MCAT, they'll usually give you the pKa value of an amino acid. But it's just kind of, if once you memorize, your acidic and your basic amino acids, it kind of makes it a little bit easier, just kind of assume. So as I said, there's 20 different amino acids. It is very, very important that you know the name, structure, functional groups, 3-letter and 1-letter codes. There's all types of mnemonics out there, but this is vital. A good portion of the MCAT is just based off of this alone. So kind of starting looking at our amino acids, we're starting off with the nonpolar side chain groups, or the nonpolar amino acids. And the very first one to know is glycine. In this one, the R group is just a single hydrogen bond. So they determine this amino acid as achiral. Very important to remember. And then our next one is alanine. Its R group is just a methyl, nothing special. Then you have valine. So valine to me looks like a V, and that's how I've always remembered it. Leucine's R group is a little bit more, it looks like valine, but with an extra carbon. And then isoleucine starts to get a little tricky, but you have a kind of this like L-shaped thing going on. What I think is very important for you to remember is these structural formulas. Everything here on the right isn't as important right now, but we'll come back to it in a later section. So kind of looking at some more with the non-polar side chains, you have methionine. What's interesting about this one is that it has like a sulfur moiety within its chain. Proline, it's interesting. It's like a pentagon ring that kind of like comes back on itself. Phenylalanine, it's just like a benzene ring. And then tryptophan, you see like kind of like this hexagon benzene ring and like a pentane kind of attached to it. And now moving on to our ones with polar side chains. So an easy way to remember that these are polar is just to determine like, the type of group that is attached. So serine, it has a hydroxyl group, very polar group. Furanine, also, polar group, hydroxyl. Asparagine has this kind of amide thing going on. Glutaminine is the same thing. Tyrosine has another hydroxyl, and then cysteine has a sulfhydryl group. And then moving on to some more polar ones, but charged polar. You have lysine, which has this amino group way out there. You have arginine. You have histidine. And then you have your acidic ones, like aspartic acid and glutamic acid. And kind of like how I was saying before, I think, at least in a starting point, it's important to know the structural formula. Again, the pKa values will come in handy later on, but what's best to know is the structural formula, the three-letter name, and the one-letter name. So moving on, we're starting to talk about isoelectric point. And this is the pH at which the net charge on the amino acid is neutral. Solubility is minimized and the amino acid / protein will not move in an electric field. Kind of for a lot of like diagrams, you see this described where the isoelectric point is in between the pK1 and the pK2. Now, what I want you to know, or at least what the MCAT wants you to know, is that in calculating the isoelectric point, you'll need the pK of the amino group and the pK of the carboxylic group. So kind of how I told you before, right? The pK of the amino group is 9, and the pKa of the carboxylic group is 2. So that's 11. And then 11 divided by 2, that's roughly like 5.5. So when it comes down to neutral amino acids or nonpolar, uncharged amino acids, now in determining the isoelectric point of an acidic amino acid, so let me draw it out here. You are going to use the pKa of the R group and the pKa of the carboxylic group and divide that by 2. And now for a basic amino acid, you're going to use the pKa of the R group and the pKa of the amino group and divide that by 2. Does that make sense? So kind of moving forward, let's determine the PI, or the isoelectric point of lysine. And sorry, this is a horrible image, but over here we have our numbers. So these are the values for lysine, and its R chain, or its R group, is 10.54. So what do we remember lysine is? Lysine is a basic amino acid. So we are going to be using the pKr as well as the pKa for amino group. So that means 10.54 plus 9.06 divided by 2. And just for the sake of time, we can say that's 19.60 divided by 2. And you can tell I am not good at math. So this is going to be about 9.8 for the PI. See, just that simple. All right, now moving on towards amino acid structure, or kind of the sequence. You first have primary structure, which is just kind of the sequence of amino acids in a polypeptide chain. And then you have secondary structure, which kind of looks like this alpha helix, similar to DNA. And then tertiary structure, which is composed of like one complete protein chain. And then your quaternary structure, it's like 4 separate tertiaries put together. But we'll go into a little bit more detail about that. So one very important thing that I want you to know is that in terms of making the primary structure, you need to do something called a peptide bond reaction. So here kind of like shows you what's going on. So that carboxylic group of one amino acid is condensed onto the amino group of another. And this is a dehydration reaction, so you're losing water in the process to form this bond. But if you wanted to reverse this, you would have to add water, or it would be a hydrolysis reaction. So it's very important to know that peptide bond formation is a dehydration reaction. And to reverse it is a hydrolysis reaction. So now secondary structures, these are local structures with repeating favorable angles. Don't need to know much about that, but they're stabilized by hydrogen bonds along the protein backbone. That is the very most important thing that you need to know about secondary structure. Now, the different types of secondary structures include the alpha helix, as shown kind of here, and then you also have your beta strands, kind of shown here. The only thing that I really want you to remember from this is that there's hydrogen bonds between the backbone. You see this and that. That's the most important thing to get out of secondary structure. Alpha helices are just one form of secondary structure, and beta strands are another. And beta strands can kind of form like in 2 different ways, one being anti-parallel and the other being parallel. So these are just kind of more images displaying that, but nothing high yield here. Okay, and then kind of moving on towards like tertiary structure. Tertiary structure is a 3-dimensional shape of protein. It kind of involves hydrophobic interactions, acid-base salt bridges, and disulfide links. And one of the big things that I want you to know here is the disulfide links. So disulfide links look something like this. And one of the proteins or the amino acids that can do this is cysteine, because cysteine has that sulfhydryl group. Other than that, tertiary structure, just remember hydrophobic, salt-base bridges, and disulfide bonds. And then quaternary structure, it's just an interaction between separate subunits of a multi-subunit protein. It's very similar to tertiary structure. And some of the proteins that kind of follow this quaternary structure is like hemoglobin, which is very important for oxygen transport in the body. So we've come to the end of our lesson plan, but we have 5 practice questions just to see if you kind of understood some of the concepts that were explained today, and I'll go through them with you. So number one, at a pH of 7, the charge of an aspartic acid molecule is what? So I guess the first thing to point out is that aspartic acid is an acidic molecule, meaning that at neutral pH, like a pH of 7, it will have a negative charge. Right? So kind of going back to the C, right? And the R group, but we know it's acidic. And we know this is 9, and we know this is 2. But you can assume that this is probably less than 7. So at this point, you see, okay, I have 2 negative charges and a positive charge. So 2 negative charges and a positive charge must mean that this aspartic acid molecule carries a -1 charge. All right. So which of these statements about peptide bonds is true? A, their formation involves a reaction between an amino group and alpha carbon. B, they are formed by dehydration reactions. Or C, they are primary bonds involved in secondary structure. So the answer is B. They are formed by dehydration reactions. And to reverse them would require hydrolysis. 3. Tertiary structure is most likely to be held together by A, hydrogen bonds, B, disulfide bonds, or C, peptide bonds. Tertiary structure involves hydrophobic interactions, salt-base bridges, or disulfide bonds formed between cysteine molecules. Hydrogen bonds would be the best thing for secondary structure, and peptide bonds would be the best thing for primary structure. Number 4, a particular protein is known to be able to cross the cell membrane. Of the following, which of these amino acids do you expect the protein to be rich in? So this requires a little bit of background knowledge, but what I can tell you is that the cell membrane is very hydrophobic and doesn't allow positively or negatively charged molecules to pass it, but it does allow for hydrophobic molecules to pass it. And so what we know is that amino acids are put into like a lot of different groups, one being nonpolar hydrophobic. So in this list of options, I think our most hydrophobic would be valine. And then number 5, in aspartate, the pKa of the side chain is about 3.90. Assuming that the pKa of the carboxylic and the amino groups are 2 and 9, the pi of aspartate is closest to. So this is another equation where we would use our carboxylic pKa plus the pKa of the R group and then divide it by 2. And so that gives you 5.90, divide that by 2. Again, not the best at math, but it should be about 2.95, which is answer C. All right, great. And that is the end of our lesson. Thank you for watching and hope to see you with the next lesson plan.